So today we look at sample questions for week 12 of the NPTEL course Experimental Biotechnology. So the first question is the biosafety level is recommended while working with Mycobacterium tuberculosis H37RV is dash. Option A biosafety level 1, option B biosafety level 4, option C biosafety level 3, option D biosafety level 2. You can take a couple of minutes to think about this and then you can answer this. Okay, Lata, can you hear me? Lata Kumari?
Yes, sir.
Yeah. So, do you understand the question? Do you have any comments on the question?
That is C, biosafety level 3.
Okay. What are the other biosafety levels, and for what kind of organisms do we work with different biosafety levels? Do you have an idea about it?
Steps for 2, it's compatible distance.
Sorry, can you repeat?
Biosafety level 2 is for compatible distance.
Compatible strains. Okay. What are those?
What are those, sir? Hepatitis virus, staphylococcus.
Can you work with viruses on BSL2 labs?
For some time, sir.
So it depends on what kind of virus, whether it's pathogenic or not. Okay. Can you name a virus which you can work with in BSL2 labs?
Staphylococcus
Staphylococcus is
pyogenes.
not a virus. Staphylococcus is
No,
a
sorry.
yeah, yeah.
It's I have worked with the staphylococcus. So yeah,
Staphylococcus aureus is it?
GG. Yes,
Okay. So that is your skin disease causing microorganism. Fine.
yes.
Yes. And by safety level 1 and 4, any idea?
For 4, it's a smallpox virus, if
Smallpox.
I'm not wrong.
Okay. For sure, I know that Ebola viruses you can work only with
Ebola.
biosafety level 4. And what
Ebola
is the
virus.
kind of protection that you get in each levels? Are you aware of that?
So once again, you can repeat your
Yeah,
question.
yeah. What is the different levels of protection that you have in different biosafety levels? Like for example, in biosafety level 1, what are the equipments that you wear? What are the things that you wear? Do you wear a lab coat? Do you wear gloves? Or do you wear a PPE suit and stuff like that?
Biosafety level 2, we have to wear PPT suit, a lab coat also, even shoes also.
Right.
We have to wear, sir.
Okay. Okay. Right. So, good. So, if you see, in general, in biosafety cabinet, okay, this is just a depiction of how biosafety cabinets look like. Uh, Here, what you see is this class 1, class 2, and class 3, which also represents the kind of cabinets that you have in different biosafety levels. Like, for example, in class 1, you work with basic microorganisms like E. coli, whereas in class 2, you work with Staphylococcus aureus, like you had mentioned. In staff 3, it is difficult. You don't have access to as much area like in class 2. Instead, you have these rubber gloved regions within which you can access your stuff. So even uh, for many viruses and even for MTB H37RV, which is very pathogenic, they use this. And for class 4, people use PP suit with supplied oxygen and stuff. So within the chamber itself, they are entirely covered just to ensure there's no leakage. The next question is the number of electrodes present in a pH meter is option A 4, option B 1, option C 2, option D 3. You can take a couple of minutes to think about this and then we'll discuss. Why
Silver
is it 2?
chloride.
Sorry?
Silver chloride.
Silver chloride. Can you be a bit louder? Silver chloride. Is that what you said?
Sorry, sir.
No, no. Can you repeat
Yeah.
it?
Silver chloride, sir.
Okay.
Chloride.
Okay. Silver chloride. Yes. And the other one?
Mercury and chloride.
Okay. So, what are these two different? What is the name of these two different electrodes?
One is glass bulb,
Yes. That's the
and
one that we
second
see
one.
on the left. Yes.
Yes, sir.
Yes. And the second one is?
Just forgetting the name.
So the second one is your reference electrode. So based on the
Okay.
reference electrode is how you get to measure your actual pH. Right. The next question is how many grams of glucose will be required to make 100 ml of 0.1 molar glucose solution? Molecular weight of glucose is 180 gram. Option A 18 gram, option B 0.18 gram, option C 180 gram, option D 1.8 gram. You can take a couple of minutes to think about this and then tell me how you calculated this if you know the right answer.
Molarity equal to weight by molecular weight into 100 into volume. Sorry.
into volume. Okay. Okay. Wait. Let's consider this as
One
a.
minute, I have to
Let's
check.
consider. Uh, what is on the numerator? Molarity. What in the formula for molarity? What's on the numerator?
Numerator?
Numerator. Yes.
Mhm. 1 minute, sir.
Yes.
Sir, on numerator it's given weight by molecular weight
What is
into
that? What does that quantity tell you? Weight by molecular weight?
number of moles.
Yes. Okay. So that's on the numerator. Okay. And the denominator?
Volume, sir.
Volume in
Liter.
in a
Either in
liter.
liters or
No. So ideally you convert whatever volume that you calculate for from ml or any other unit to liters. Yes.
okay, okay.
Yeah. So molarity is number of moles per liter of solution.
Yes, sir. Yes, sir.
Yes. So how will you get 0.18 gram?
By mistake, sir. By
Okay.
mistake, I have said
Okay.
1.1 gram.
Okay. So if you look at this, it would appear like 0.1 molar on the left hand side equals x divided by 180 divided by 100 by 1000. So when you cross multiply, you will get 1.8. Right. The next question is, a PhD student is facing problem of acrylamide polymerization even when he is allowing polymerization reaction for 30 minutes at room temperature. Which of the following components is most likely to be missing from the cocktail? Ammonium persulfate, trisacil, STS, and glycine. Take a couple of minutes to think about how this polymerization happens, and uh, so in this acrylamide polymerization, you have to before we get on to the right answer for the question, you can tell me how this is initiated, what initiates it, and how is it propagated. Take a couple of minutes to think about this, and then we'll discuss.
Polymerization.
It's okay. In ammonium persulfate, how does it act as the initiator? Do you have any idea? How does the polymerization take place?
Sir, acrylamide, both the acrylamide helps combine with the APS, and when we add the timide, it helps in catalyst, and it forms free radicals, and then it get bind to each other.
Yeah, so here your persulfate is the free radical that is needed. So without the persulfate free radical, you cannot have polymerization. In each polymerization reaction, you have a free radical which is an excess electron or a which has unequal electrons or unpaired electrons, and that is what helps you in the polymerization reaction. And the other options here, tris, STS, and glycine, have nothing to do with polymerization because tris is your buffer, STS is your detergent, and glycine is a component in your buffer which actually helps in moving your protein down the gel. Good. The next question is, a complex protein mixture was separated by STS page and the RF values calculated for 4 protein bands present in the mixture are given below. Protein 1 is 0.33, protein 2 is 0.5, protein 3 is 0.16, and protein 4 is 0.83. The protein band closure closest to the well is dash, and 4 different options are given here. So before we go to this, take a couple of minutes, think about what RF is, how RF is defined, and based on RF, try to make on the gel, which band will be on the top, which will be the second top, third top, and the last one. Okay, so take a couple of minutes. You can start by defining what RF is. Okay, do you know what RF is?
So relative mobility.
Okay.
Or
What does that represent?
what is the
What does it represent?
molecular weight of protein?
Does it really represent the molecular weight of protein? Is it actually calculated only for proteins? Does it have any formula?
Sir, RF means retention factor, sir.
Yes, retention factor. Yes. And
I mean distance traveled by substance by distance traveled by solvent.
okay. Here the solvent would be in. Okay, good. So in a case where we have STS page, what will be the appropriate solvent?
Sir, we use many things, sir, like different types of buffer, stacking, resolving gel.
Oh, okay. No, no, that will not be the ideal case here. So let's say you run the gel, but you don't run the gel till till full, till its entirety. Okay. In that case, let's say you run only till half of the gel length. Now, when you calculate the retention factor, what will be in your denominator? Let's say the gel length of the gel is 10, but you run the gel only till about 5 centimeters. Now, what will be in the denominator for your retention factor when you calculate? Do you get my question?
Yes, sir, I got it.
Yes. Is there something that you'd add to your samples that helps you to calculate the retention factor?
Uh, could you repeat, sir?
Yeah, yeah. Is there something that you add to your protein that helps you calculate the retention factor on each lane?
Sir, we like tracking dye. We can, by using it, we can find where it is.
Yes. So your tracking dye is the solvent in an STS page, according to your definition of retention factor. So retention factor typically tells you compared to your tracking dye, how much has different uh, what do I call it, metabolite or molecule of interest has traveled compared to the tracking dye. So like you had mentioned, building upon it. So the retention factor is the ratio of the distance traveled by your molecule of interest in a particular lane compared to the distance traveled by your tracking dye. In most cases, we switch off the gel once the tracking dye reaches the bottommost region of your gel. So ideally, the denominator is equal to the length of the gel. That is what is being traveled by your tracking dye. Good. So now, if that's the case, proteins that have higher molecular weight, what will their retention factor be? Will it be low or high? Did you get the question?
Sir, it's a little bit confusing.
Oh, no, no issues. Did you understand? We agree that tracking dye will be our denominator. The distance traveled by the tracking dye will be in our denominator, right? We agree on that.
Yes.
Yes. Now, let's say there is a protein uh, that is of higher molecular weight. If it's of higher molecular weight, what will its retention factor be? Will it be higher or lower? That is the question.
Sir, is it higher?
Why will it be higher?
By just I took the random number, I just divided by it, so I got like.
No, no. Why will you take? Okay, let's say a protein. Okay, on a gel, when you say okay, let's consider two proteins. One protein is 80 kilodalton, the other protein is 20 kilodaltons. Okay, and the tracking dye has moved for a distance of 10 centimeters. Now, among the 80 kilodalton and the 20 kilodalton proteins, which of them would have traveled the most? Is it the 80 kilodalton or the 20 kilodalton protein?
A 20 kilodalton.
So, if the 20 kilodalton protein has traveled the most, the retention factor for the 20 kilodalton protein, will it be higher or lower?
Higher, sir.
Yes. So, proteins that are of lesser molecular weight, they travel farther. Because they travel farther, they'll have a larger retention factor. Whereas proteins that are of higher molecular weight, they travel for a relatively lesser distance. So, their retention factor will be less. Was that clear?
Yes.
Yes, yes. That is why here, even in this case, the protein band that is closest to the well has the lowest retention factor, which is 0.16, and that is protein 3. Was it clear?
Yes.
Yes, sir.
Yes, good. Okay. The next question is agarose acrylamide composite gels are used to analyze dash antibodies, large multimeric protein complexes, dimeric proteins of size 150 kilodalton, small proteins of 44 to 40 kilodalton in size. Okay. So before we go to the right answer for this question, what you can do is you can tell me what are the different kinds of gels that you're aware of, and typically what is the biomolecule that you run on those gels. You can take a couple of minutes to think about this and then we'll discuss.
Sir, on agarose gel, we can run nucleic acids, and
Okay.
acrylamide we run proteins.
Okay. Uh, on agarose, you said we run nucleic acids. What is the typical molecular weight? Or okay, first tell me what is the kind of nucleic acids that you run on agarose gels?
DNA.
Yeah, yeah. Molecular weight or base pairs, anything of that sort that you know? Because here we are trying to get an idea about the size of the protein or whatever biomolecule we are interested in. So, based on the size, do you have any idea? The length of the DNA or the number of base pairs or anything of that sort?
Like 100 base pairs to, like, I remember like 100 base pairs to 25 kilo bases.
25 kilo base pairs.
Like 100 base pairs to 25 kb.
25 kb. Okay. This is on an 1% agarose, is it?
Yes, sir. 0.8 to 1%.
0.8 to 1%. Okay. So, what is the typical molecular weight that you use for 100 base pairs? Of what will be the typical molecular weight of 100 base pairs of DNA?
Sir, one base pair weight is 617 grams.
Grams.
You asked what did
No,
you
no, is
ask,
it
sir?
in grams? Just check the unit.
Yeah, I said grams per mole.
Is it not in Daltons? Okay, fine.
Okay, you're asking Dalton, sir.
Yeah,
Okay,
yeah, yeah,
okay.
Daltons. That'll because we usually when we calculate the mass of a protein, we mention it in kilodaltons. We say amino acids have each amino acid is approximately 110 daltons, and then we calculate depending So, yes, you can mention that in Daltons.
So 66,000 Daltons.
Is it? Is it 66,000 or 660 Daltons for one base pair?
One base pair is 660, for 100 base pairs is 66,000.
Yes. So 100 base pair DNA is 66 kilodaltons, right? Your average protein is of the size of 40 to 50 kilodalton. So you say for DNA of the molecular weight of 66 kilodaltons, you use about 1% agarose, and that is the minimal length of DNA that you more or less use. If you want to use lesser than that, you can use acrylamide. And so typically, because of this molecular weight thing, we use single stranded RNA with uh, polyacrylamide gels, double stranded DNA with agarose, and proteins with uh, STS pages. Now the question is, when do we use agarose acrylamide composite gels? Uh, Now here, other than normal proteins, you have something called as multimeric protein complexes. This means that you have multiple chains for a given protein or protein complex. By protein complex, I mean those functional assemblies that you see like in the electron transport chain, which are physically connected together. I mean, they associate some in majorly they associate in their oligomeric status, and then through non-covalent interactions, they are assembled on a single site. So for these are on the order of megadaltons, which is very high, and we typically if you want to know whether your assembly has all the proteins involved in the protein complex, what one would do is they would run this on a gel and then expect the right molecular weight. So in those cases, it is said that you can use these agarose acrylamide composite gels. But for normal proteins like dimeric proteins of this molecular weight, like they have mentioned, its HTS space is more than sufficient, and even for antibodies. Was that clear?
Yes, sir. Sir, I have a doubt.
Yeah,
Like,
yeah.
if we use less than 3% of acrylamide
Yes.
gel,
Yes.
does it form?
Does it form as in? I didn't get your question.
I mean, say, if you use like concentration 3% concentration of acrylamide in the HTS space, does
Oh,
the gel
3%
form?
acrylamide. Okay.
Yes, sir.
In
Does the gel
STS
forms
phase.
or not?
So, okay. Before this, I would ask a question. What is the concentration of acrylamide that you use in stacking solution? So your acrylamide gel has two phases, right? Stacking and resolving, right?
Yes, sir.
Yeah. So in your stacking, what is the concentration?
Depending upon size of the protein we use, no, sir. Like,
Is it?
generally 3 to
Don't you
5.
have a defined stacking? And resolving gel is what would vary, because that is where your proteins get separated. In the stacking, you ensure all the proteins are uh, at the same starting point. They are sandwiched between the two layers, so that in the
Yes,
resolving they can separate. So the stacking gel has a typical defined concentration, right?
5%.
Yes.
Like,
So if your stacking is 5%, can your resolving be 3%?
uh, yes, sir.
Can it be?
It can be.
But why will it be? So what I'm trying to tell you here is, in your stacking gel, you are are trying to sandwich it. Of course, pH plays a role, but one thing that you majorly need to consider is in a stacking gel you won't get uh, in the concentration of stacking gel you won't get ideal separation. So I am not aware personally of anyone using 3% gels in resolving gels. We always use anything above 8%. At least for the proteins that I work with and the RNA that I work with, we use anything above 8%. I've never personally used 3% resolving gel. You would get this gel. Physically, it is possible to make that gel. Something will travel through it, but the resolution with which it separates will be uh, very fast. It's not going to. You might lose your protein of interest in that unless your protein is very large. I have not personally worked with such a concentration, but it is physically possible to make such a gel, although it will be fragile.
Yes, sir.
Okay. The next question is the distribution coefficient values of four proteins are given below. Protein A 0, protein B 1, protein C 1.5, protein D 0.5. If the mixture of these proteins is loaded on gel filtration column, which protein will come out first from column? A. Protein A, option B, protein B, option C, protein C, and option D, protein D. Take a couple of minutes to think about this. You can start with mentioning what distribution coefficient is and in the context of a gel filtration column, and then we'll take it from there. distribution coefficient is and in the context of gel filtration chromatography?
So the distribution coefficient is how much solute is distributed between the mobile phase and stationary phase.
Okay. So in a gel filtration chromatography, what is your mobile phase? What is your stationary phase?
Uh, Stationary phase is the pores of the beads,
Huh. The beads, yes.
and mobile phase is outside of the beads.
No, no. Isn't your mobile phase something that is flowing?
Yes, sir.
So is it not the solvent which is carrying your protein of interest?
I mean, solvent outside the beads. I mean, there's a space between the beads, no, sir.
Yeah, yeah. But
Solvent will be
but isn't
between.
the solvent the mobile phase here? Leave the space. The space is filled with some buffer.
Yes, sir.
You might ideally always what we do is the we call it the equilibration step where we run the column without injecting the protein, but with the buffer in which we want to elute out the protein, so that the column is already having the conditions necessary for the protein to fold well and be stable. So in these cases, isn't the mobile phase the solvent or the buffer here?
Yes, sir.
Yes. So it's not the space. Correct. So in gel filtration chromatography, your stationary phase is your bead and your mobile phase is your buffer. Right. Now, uh, yeah, coming back to KD, you told it's the ratio of partitioning of your uh, solute of interest between your stationary phase and the mobile phase. Now, compare these for these four proteins with KD equal to 0, 1, 1.5 and 0.5. Can you tell me anything that you know regarding how they can preferentially bind to the beads or elute with the solvent?
So, like I remember, if the KD is zero,
Hmm.
the fragments, I mean, particles are too large, so it doesn't get inside the bead, so it gets elute first.
Okay. Okay. And what about other cases?
If it is greater than zero,
Hmm.
then it may enter into the beads
Hmm.
and elute later, depending upon the size.
Okay. And can it be greater than 1? Because for protein C it is given as 1.5.
Don't know what happens if it is greater than 1.5.
No, no, you just
I'm sorry.
think about the ratio. Just think if KD can be greater than 1.5.
So it may interact with both the phases.
It is a part of one phase now. So from what I know, I don't think your KD can be greater than 1.5. It should be somewhere between 0 and 1, where 0 means uh, it is not at all binding to your beads, and 1 means it's fully bound to your beads. is why even in this
So maximal is one, right, sir?
yeah,
That's it.
maximal is one. That's what I just wanted you to think about it, which is why I asked if the option protein C is even physically possible, because this is a ratio, right?
Yes, sir.
It's like, let's say you have I mean, in a ratio, when you take the denominator as the maximum, you can't have it more than one. So uh, that's why this is between 0 and 1. And when it doesn't bind to the bead at all, which is kd equal to 0, then it will elute first, right? Is it right? When it doesn't bind to the bead,
Yes.
then kd will be 0, it will elute first. That is why protein 1 with distribution coefficient 0 will be the first to come out of the column. This just tells you the typical textbook definition that the higher KD means it has greater affinity for stationary phase and it will be retained longer. And lower KD means the compound prefers the mobile phase and it will elute more quickly. But this value, when we change the system from proteins to small molecules and partitioning between two different kind of solvents instead of a stationary phase and a mobile phase, there it can be greater than 1. Like, for example, you have cases of octanol water systems where compounds can preferably bind to, I mean, preferentially um, be in one particular kind of solvent. You have hydrophobic solvent and hydrophilic solvent, which is your water. So when you have a mixture of these both, in some cases they do mention that this distribution coefficient or partition coefficient is greater than 1, but there it is calculated differently. It's not the ratio. Fine. The next question is, a nuclear protein is involved in regulation of gene X. The protein was incubated with gene X, which is ligand, and ran in a gel filtration column, and the chromatogram recorded. Select the correct statement from the following. Protein ligand complex have lesser retention time. KD value of protein ligand complex will be more. The peak corresponding to the protein ligand complex will show a right shift. Hydrodynamic surface area of protein ligand complex is lesser. Take a couple of minutes to think about this and then we'll discuss. incubate a protein complex and run it on a gel filtration chromatographic column. When you run it on the column, does it tell you anything about the KD value?
Yes, sir.
Does it tell you anything about the KD value? KD here can be called as your rate of dissociation, because this is K small d, not K capital D.
So I thought it's a dissociation. I mean, say partition coefficient.
No, it's not the partition coefficient here. This
Okay.
is different. Okay. Does it? It's the dissociation constant. Dissociation constant tells you the rate at which these two proteins can bind and unbind, considering that all these exist under an equilibrium. Does it tell you anything about shift of peaks? Okay. So in your chromatogram, what you see is a graph that shows you how over time you have absorbance that is changing. So if you get a peak, that is just indicative of a protein or a protein protein complex. So protein protein complex are larger. These will elute first compared to individual proteins. So usually, let's say you run three samples. One is your actual nuclear protein, the other is your gene X, the other is a complex formed by this nuclear protein and gene X. So let's say your nuclear protein is 60 kilodalton, your gene X is 20 kilodalton, and the complex is 80 kilodalton. So you will see three different peaks when you run these three. And the first peak will be of the complex because it's of the higher molecular weight. So option C that says the peak corresponding to protein ligand complex will show a right shift is wrong, because that will be on the extreme left. That will be the highest molecular weight. That will be on the extreme left. KD value you will never be able to measure from these experiments. You need an experiment. Uh, okay, depends on whether it's binding affinity or dissociation constant. So you have methods that measure the rate of association and dissociation, like surface plasmon resonance. You can use that. And the third one is hydrodynamic surface area of the protein complex is lesser. So when protein interacts with another protein, the buried surface area is what will be increasing. And because the buried, that tells you how much of water is being displaced from a particular surface. Hydrodynamic surface area talks about all the outer external ones. That gets increased, that is not decreased. So that is also wrong. The only option here is that is right is the protein ligand complex would have lesser retention time compared to your individual proteins. Was that clear?
Yes, sir.
Okay. Okay. The next is a simple textbook definition question. The parameter that defines the binding capacity of antibody to an antigen is dash. Avidity, affinity, cross reactivity, and specificity.
Sir, if it is binding for multiple epitope means where another definition will come, another meaning it will come, sir.
Yes. So in that case, what is the information that you would want to convey?
For affinity, it is a single epitope. For avidity is like multiple epitopes
How many
binding.
can it bind? Yes.
Like multiple.
Huh. Multiple, but is it of the same type or the different type? So
Different.
different.
Different. Same antigen, but different epitopes, sir.
Hmm. Okay.
Sir,
Yeah, yeah.
yes, sir.
You can repeat your question to me. It sounded simple, but you can repeat. I'll again go through it. Yeah, you can tell me.
I mean, avidity is nothing but a paratope is binding to different paratopes are binding to different epitopes. Within an antigen or different antigen, I
So
don't have no idea about it.
so let's say you have an antibody that has multiple binding sites. This avidity concept talks about valency. How many of it is binding to how many, right?
Yes, sir.
So if your antigen has multiple binding sites, and antibody also has multiple binding sites, if it interacts, you can conclude that these multiple affinities as avidity. Is that a fair statement to make?
Yes, sir.
Yes. Whereas affinity will be one to one interaction, and that is quantification. And whereas cross reactivity will be one antibody being able to bind to some other antigen of that is not of our interest, which usually doesn't happen. Whereas specificity talks about again one is to one interaction and specifics on what is binding to what. But I still didn't get your question, Sai Sampath.
Sir, like one antibody can bind to different antigen. Is that statement correct, sir?
One antibody will be raised for one antigen.
Not
The,
different antigen, sir. Like
it will be raised for one antigen within the same antigen. You so this is a physical interaction, right? You have bonds that are being formed, right? Non-covalent
yes, sir.
interactions are happening, right? Now this non-covalent interaction can be one to one or many to many. In avidity, we consider that this non-covalent interaction is many to many. You have multiple sites on the antibody that binds to multiple sites on the antigen.
On antigen, you are saying, no, sir?
Yes.
Like different epitopes are there on the antigen, right,
So
sir?
how do you define an epitope? An epitope is the smallest unit which can for which an antibody can be raised, right?
Yes, sir.
So if the smallest unit is not well defined, but if the smallest unit is an exposed patch, let's say of 10-20 amino acids that forms a particular secondary structure, then in those cases uh, the antibody will be covering all those exposed surface, right? So again, it's not
Yes,
one is
sir.
to one. So what I'm trying to tell you here is, although if you look at it at a very very low resolution, stating that this antibody binds to this antigen only, if you look at the number of interactions specifically at the atomic level when you solve the crystal structures of these antibodies with the antigen, what you will be able to identify is there are multiple different parts from the same antigen. You have multiple epitopes and paratopes that are part of each of these, which constitutively dictate the antigen antibody binding. Does that
Yeah,
make
got it.
sense? Okay.
I understand. Yes, sir.
Okay. Agglutination assay are used in dash ABO blood typing, bacterial meningitis detection, determining of antibody concentration, detection of soluble molecular antigens.
APO lead type things.
Yes. What is agglutination?
It's the interaction between antibodies and antigens.
And what does it result in?
Formation of clumps,
Right,
visible clumps.
right, correct. So, based on these visible clumps, you know the blood type. Correct. And the reagent that is used to develop an HRP based secondary antigen and secondary antibody system in a Western blot is DASH. So, before we go to the right answer for this, if you know what HRP is, you can let me know.
Sir, horseradish peroxidase.
Yeah, horseradish peroxidase. So, what is that?
It's an enzyme. It is linked to the antibody.
Hmm. So, what does that enzyme do?
Sir, when it reacts with the substrate, it gives specific color. Whether it is to know the quantification of the percentage of antigen present in the sample.
Okay, so what it typically does is because it is linked to an enzyme, when you give it a substrate, it gives luminescence. It converts it, and from the name peroxidase, you'll be able to tell that with luminol and peroxide, it will give you luminescence, right? And that can be quantified, which is what is done in Western blot. The next is what happens to the color of mammalian cell culture DMEM media when the pH of the media decreases? And four options are given here: changes to bright red, changes to blue, changes to yellow, changes to green. So you can take a couple of minutes to think about this and then tell me how the color changes and what's the actual color of DMEM media at the physiological pH in which it functions.
Sir, we use phenol red as a pH indicator in mammalian cell culture.
Okay, so what is the color of the media at physiological
It
pH?
is generally in red color, 7 pH.
Okay, okay, right. And how does the color change if it becomes acidic, and why will it become acidic?
Sir, if it becomes acidic, it's yellow. If it becomes basic, it turns purple. It's phenol red. So I said like that.
Oh no no, but
Based on
why will it become acidic, or when will it become acidic? When will the pH of that DMEM media change to become acidic?
due to CO2, I think so.
Yes. So when CO2 is released, uh, it becomes acidic because water plus CO2 makes carbonic acid, and acid is acidic. So if it's acidic, it becomes yellow. If it's basic, it becomes purple to pink. And usually at physiological pH, it's red to orange. And like you mentioned, phenol red is the pH indicator here. It's just a continuation. I So here in this question they ask what's the function of phenol red, and as we saw earlier, it's the pH indicator. Okay. The next question is, flippin staining can be used to study the phagocytosis of latex beads by J7474A1 cells. Flippin can stain the dash integral proteins of the membranes, cytoskeletal proteins, cell membrane, periplasm. Okay, so here they say that this is a fluorescent antibiotic that can bind to unesterified cholesterol, and cholesterol is present in your cell membrane, plasma membrane. So this is used to study membrane structure and dynamics. So the answer is cell membrane. The last question is, which of the following proteins prevents re-annealing of the single-stranded DNA? Helicase, primase, SSB proteins, and DNA polymerase. Take a couple of minutes to think about this, and then we'll end the class.
Sir, answer is SSB protein.
Can you
Single strand.
can you expand what SSB is?
Single strand binding protein.
Ah. So what does it do?
DNA is double helical.
Yes.
So during replication, they are separated,
Yes.
or they cannot be rewind.
What will happen
Is
if it
SSB,
rewinds?
if they rewind, then termination will occur,
Hmm. Extension
or
or
double helical.
replication will not happen, right? Yeah.
Yeah.
Yes. Hmm. So you need these single-stranded binding proteins to help the DNA polymerase to sit and as the helicase unwinds to carry out replication. Right. Good. So with this, we come to an end for today's lecture. There'll be one more session on Monday, a special class where we discuss all the concepts more or less that are covered in this course. If there are any questions, you can ask me. I'll stay on the call.
No questions, sir.
Okay.
Thank you.
Thank you.